So the second speaker is Lara Ojo from the University of Lagos in Nigeria, and she will be talking about the importance of the intronic GBA1 variant that Sonia just mentioned, identified in people of African origin. Go ahead.
Thank you very much for that introduction. Let's see. Okay, great. Good morning, everyone. Happy to be here. Thank you, Ziv, for the invitation. And like Sonia, I'm going to tell you to take it easy on me for one simple reason. My background is in clinical neurology and movement disorders, and happy to be here among you all scientists. So these are my disclosures. And for this session, what I'm going to try to do is to give you a background, an overview of the issue of genomics in Parkinson's disease. Because just like Sonia's presentation, my presentation will also be focused mainly on Parkinson's disease. Then the discovery of the RS3115534 GBA1 variant, the intronic variant, and the importance of this variant. So we've all been talking about GWASs and sequencing, and one thing that has really taken genetics of Parkinson's disease, has taken it far and made it leapfrog in the last few decades, or a decade and a half, is the GWASs. And despite the technology, despite the results, there is still a gap in terms of heritability of Parkinson's disease. And one of the major gaps that the GWASs brought out was the issue of generalization of those results to global populations. And that's just because most GWASs were based on people of Northern European descent, and as such missed all the genes that from other populations. Now, underrepresentation of Africans in research is not just about African patients, it's also about even the scientists doing the research. And you may be wondering, why should people or persons of African descent be important to genetic research? One of the reasons is because, one, they have a high level of genetic diversity, and this high level of genetic diversity is made possible by the complex population history in this population, the variation in the environmental factors compared to other populations. So we're talking climate, we're talking diet, and importantly too, the exposure to infectious diseases. On the right side of your screen, there is a very good review paper by Artoscu from Ignatius' team that looked at underrepresented populations in Parkinson's genetics research. And one of the things they brought out was the significant lack of data from African researchers and inclusion of Africans in those studies. And if you compare the percentage of Africans that are included in genetic research, usually less than 4%. However, when you look at that in the context of the world population that is of African descent, you'll know that this is a big gap, because as of now, that population is about 15 to 17% of the world population. El Sayed and from Sarah Bandesiga's team from the NIH looked at Parkinson's disease genetics and its impact and challenges with the impact of PD genetics. And in the middle, you can see what the genetics research is supposed to achieve from the bench to the bedside: identification of the variants, improvements in PD research, implementation of those research outcomes in clinical practice, usually by improvement in diagnostic methods, prognostic markers, and then new therapies for development. That's why we're here. However, on the left side of your screen, you have challenges that are peculiar to underrepresented populations, of which Africans belong. And when you think about Black Africans, I want you to have an expanded view. These are not just people living in Africa. You have Black Africans everywhere, and they face some of the similar types of challenges Black Africans will face. Also, underrepresented populations will include people from Latin America and even indigenous Americans. So these are not limited to the African population. So challenges such as technical issues. So while we think about technical issues in terms of lack of infrastructure in Africans, there will be the same technical issue when you're talking about a Black African who is living in the US, for instance, because they will have lack of access to that infrastructure. Issue with data analysis, so lack of the skilled personnel to even make the diagnosis and do the genetic research. Again, that will be peculiar for a place like the South Africa. However, when you think about the Black African or the African admixed, they also face these issues because there is a lack of access to places to the specialized clinics where these tests are more likely to be offered. Again, limited access to PD research. Black Africans are less likely to participate, and even Black Africans less likely to participate in clinical research and even genetic research because of the biologic sampling involved. And while at the base, as we can make it very simple and say this may be due to a low level of education, sometimes it's not about the education. It's about a low level of awareness. It's about historical mistrust. It's also about the fact that clinically they are less likely to have access to places where they are informed about genetic research or even clinical research taking place in Parkinson's disease. And then the cultural barriers, and you'll see why all these are important when we get to the importance of the variant, and then we get to the issue of cost or funds. So it's important to bridge this diversity gap, not just to be politically correct, but to improve the understanding of the biology and pathogenesis of Parkinson's disease by revealing new mechanisms, by giving new insights. More importantly, prevent underestimation of population-specific genetic risk factors. So if we look at risk prediction scores, many of them are based on the variants and the factors from people of European ancestry. So these scores will not work for African population and persons from Black African ancestry. Understand the interplay between those variants and environmental factors that are not the same globally, and then development of adequate precision and personalized diagnostics, prevention, and treatment. So Africans or persons of African descent or persons of African origins have an important role to play in genetics, and that's just because they display admixture signals in their genomes. One of the other reasons that they must be included in genetic studies is because they need to be factored in, because the assumptions of a homogeneous population structure that is more likely to be made when you're dealing with an European population will be violated with an African population, and that's because they have more haplotypes, they have lower levels of linkage disequilibrium, and they have just a more complex pattern of population substructure. And just to reiterate why inclusion is important, the GWAS catalog, though it's a bit old now, of 2021, showed that, and this is just the GWAS catalog, not peculiar to Parkinson's, approximately 2.4% of participants in large studies were of African origin, and they were mainly African admixed people. To put that in context, they accounted for about 7% of the disease-related genetic associations that were being found. So this brings to the fore, it reiterates why we need to include more people of African descent in genetic studies going forward. So I've just given you a background as to why this study, the impetus for this study. And this is the paper that published the intronic GBA1 variant, and this was the outcome of a collaboration from the International Parkinson's Genomics Consortium Africa, the Nigeria Parkinson's Disease Research Network, the NIH, 23andMe, and ASAP. And we this took place in 24 institutions, African institutions, Nigerian institutions to be precise, and 4 Black PD sites, and included 2 cohorts. So we had the African cohort that was made up of a Nigerian cohort and then an IPDGC Africa cohort, and that accounted for about 1,000 plus patients. And we had the IPDGC cohort that also accounted for about 300 patients. Then we had the African admixed cohort adding to the mix. Meta-analysis revealed the GBA1 variant that was identified as a novel risk factor in persons of African descent. And for comparison, you can see that the European, the GWAS, the meta-analysis on the GWAS from persons of European descent did not get a hit in this at the GBA1 locus, and instead, as we all know, is picking up a hit on LAC2. Surprising, it's a non-coding variant, and it's high risk for people of African descent. There's almost a 1.5 times risk in heterozygous carriers. For the homozygous carriers is more than a 2% risk. And half of all Nigerian Parkinson's disease cases actually carry the variant, and each allele results in at least a 2.5 year earlier age of onset of Parkinson's disease. As a follow-up to that, we were trying to look at the phenotype of that particular variant, especially because it was a non-coding variant as opposed to the other variants that had been discovered for GBA1. So we looked at the association of the variant with REM sleep behavior disorder in Parkinson's disease in Nigerians. And for this, we looked at approximately 700 Parkinson's disease cases and 776 ethnically matched controls. Again, when including persons of African descent in studies, be they clinical or genetic, it's also important, if you are going to match them for controls, to match for ethnicity, because Africans differ, Nigerians differ, and we have more than 50 ethnic groups in Nigeria, so we had to match each case for the ethnicity, again, just because of the diverse genetic background. As expected, we found that RBD, for this study, we used the RBDSQ, which is a screening questionnaire, to diagnose RBD and not polysomnogram diagnosis. And as expected, we found out that the RBD was more in the PD than the controls, about 28% in the cases versus 6% in the controls, which is not unexpected. We identified that the GBA1 risk variant is associated with possible RBD in individuals of Nigerian origin And that association persisted even after adjusting for the Parkinson's disease status, which suggested that although the variant was non-coding, it may have the same downstream consequences as coding GBA1 variants. So this brings me to this paper by Cornelis, and if there are any questions here, I think he's in the audience. He will be able to. So this looked at trying to understand the function of the variant, and I think the presentation from Centogene also mentioned or went through this yesterday. And using long-read sequencing, CRISPR analysis, what was found was that the risk variant disrupted the branch point, leading to missplicing, and that missplicing caused an apparent increase in GBA1 RNA. And what was the net result of that? Reduction in GBA1 protein and then reduced glucocerebrosidase activity, which is highlighted in the figure to the bottom on the right. So why is this finding important? It's important because it introduces another pathogenetic mechanism that is peculiar to a specific ancestry and population. And this is important when you are thinking about preventive strategies, when you are thinking about drug development strategies. These need to be kept into consideration. It establishes a foundation for exploring the possibility of personalized interventions. When we talk about precision medicine, we can't be precise if we're applying strategies from one ethnic group to another where it may not be applicable, especially through clinical trials, specifically targeting this population. I have forgotten the name of the presenter for Centogene yesterday. He almost fell into the trap set by Ziv when he was asked if he would include people with this variant in clinical trials. And I think he dodged the bullet and just said yes. And the question is, why not? So they will be included in clinical trials, and they should be included in clinical trials. Now, I get the subtext for the question, if I may deviate a bit, and that's just because I'm sure people are thinking, oh, if there is, or if there are challenges with this specific population, if there are challenges even doing the research, if there are challenges even assessing care, how do we develop a medication that targets this particular gene in this population? But the truth of the matter is the aim of everything we do, the aim of translation science, the aim of GWAS studies is to give us more information and ultimately to improve health outcomes of all persons living with a particular disease, and in this case, Parkinson's disease. And it is important when you realize you have an ancestry-specific variant or risk factor, because that also needs to be taken into consideration when planning those clinical trials, when planning those clinical studies. Some of the reasons clinical trials fail is just because the population being targeted wasn't put into focus when the team was being assembled. So if you know you are recruiting patients or you need people of a particular ethnicity in your research, it doesn't augur well when nobody on the research team looks like them, when nobody on the research team talks like them, when nobody on the research team can't speak to their challenges. So these are some of the things we need to put into consideration when planning studies and when planning clinical trials, especially those trials that are planning to include a more diverse population. It underscores the scientific and ethical imperative of including underrepresented populations, and underrepresented populations are not just Black Africans, Latin Americans, Indigenous Americans, even women with Parkinson's disease, an underrepresented population. It adds to our understanding of the potential role of non-coding variants in the risk for Parkinson's, and highlights, I think I've mentioned this, the notion that findings cannot be generally extrapolated to other populations. And it also provides an impetus for refining our polygenic risk scores for Parkinson's disease prediction in persons of Black African ancestry. This brings me to my take-home messages. So we need to demonstrate how do we improve the recruitment and retention of persons of Black African ancestry in any clinical study, any clinical trial, no matter where they are taking place. You must demonstrate clinical utility in a local context, and this requires a lot of trust building. As I alluded to earlier, there's a lot of historical mistrust when it comes to participation of Africans in research, and this may require having somebody, an engagement with the community physician who interfaces with them and who can actually change their misperceptions regarding research. Community engagement and awareness is very important and crucial, and that's because the success of translational science depends largely on the availability of quality data, and to get that you do need individual and community cooperation. And then the development of precision. So we talk about precision medicine on an individual level. We also need to start looking at precision public health strategies. So it's not, public health will not be a one glove fits all at the end of the day, because the interventional strategies that apply to one population may not really apply to other populations. And at this point, I'd like to say a thank you to my team. I don't have a lab, but on this upper part of the slide, those are the faces of neurologists in the Nigerian Parkinson Disease Network. Would like to say a big thank you to our funders, Aligning Science Across Parkinson's, Global Parkinson's Genetic Program, the Michael J. Fox Foundation. A special shout out to our collaborators at the UCL, the NIH, and more importantly, thanks to our study participants and their families. Thank you for attending this session, and thank you for listening.
That was great. We have time for some questions. Zip has a mic first.
Thank you very much. This was a very good and interesting and important talk. So I have a comment and a couple of very quick questions. One, I did set a trap and it dodged the bullet, like you said, but the answer I was actually looking for was it might make this population maybe the even most important one for clinical trials, because we are in trouble in the sense that GBA is hard to screen. There's not enough clinical screening initiatives, and there's a lot of competition on the patients because there are many companies now going into phase 1, phase 2, phase 3, hopefully soon trials. So I think this population is actually will be crucial for that, because it provides a very large resource, pool of people that can participate in clinical trials and benefit from it, hopefully. So that was what I was aiming for. I have two quick questions. One, is there polysomnography ability in Nigeria?
It's very scarce, so maybe in the private setting, none of the tertiary centers have the capacity for it. So maybe one, if we're talking it's available, you're talking one center in the whole of Nigeria.
Okay. And my second question, I assume you are, but are you starting to look at the effects of cognition and things like that in this population?
Yeah, so that was one of the gaps that we couldn't actually publish with that Lancet Neurology paper, because two things: we didn't have enough cognitive data to do that analysis. The second thing, again, which goes to why sometimes people don't, persons of other populations don't take part in research, is because when we started the recruitment, we just found out that what we took for granted, the MOCA, which is your straightforward screening bedside office tool for cognition, was performing woefully in the population. It was actually an eye-opener for us, even as neurologists and clinicians. And at some point, we had to add on another screening tool that was tailored towards our population. So again, that is something you do need to take into consideration when you are recruiting persons from other populations. The MOCA is just abysmal when it comes to a population that has a high rate of lack of formal education.
Thank you for an excellent talk, Roy. I see the group of the physicians that helped recruit, and I had the quick question. The first quick question is, how many languages did you have the consent form to? Like, because it looks like a huge effort, and Nigeria is a very diverse country, right? So, how many languages is there the consent form in?
Okay, so that's a very good question. So the good thing with Nigeria is that the official language is English.
Okay.
So our communication, everything is in English. So even the person who lacks formal education can speak a variant of English known as the Pidgin English. So it was a bit straightforward. We only had the one.
And the follow-up question on it is whether have you started or are you considering returning the result of the GBA variant to people? And what would it take to help you do that?
Okay, so great question. So one of the conversations that started just about the time this variant was discovered and published, and that has become a big conversation now, is the return of results. And return of results is a very big thing. Number one, you must be able to inform on the implications of the variant, both to the patient and to the family. And again, it's about having genetic counseling pre and post. So one of the things we were scared of when it came out was, or when we first started, was would people be interested in even knowing? But we were surprised. Even before results came out, we kept getting questions. Was anything found? If anything was found, what is the relevance of what was found? And I think that is one of the reasons why most consortia, most research groups are now coming together to have a formal way to return results. So the same way you are considering how to formally return results of research participants to them in advanced countries is the same way we are thinking about it. Now, what do we lack? We lack genetic counselors. Genetic counseling is not something we have, so it's still going to be the same neurologists that will probably need some crash course on genetic counseling and then ability to deliver the results when the full implications and the wider implications of that variant are known.
Okay, thank you, Laura. We have to get moving, but you can catch up, Laura, at the break if you have any questions. So.